So this is work done by my two colleagues in the lab, Dr. Maria Teresa Fulciniti and Yan Shu. It focuses on an interesting molecule that we identified, which is present on chromosome 17p. It is called YWHAE, or its other name, which is a little bit better known, is called 1433 epsilon. And the reason it is very interesting is because this molecule is known as a chaperone, and it has number of activities described, not specifically this molecule, but there's a whole series of 1433 proteins that are involved in various drug resistance and sensitivity, etc. And their chaperone function makes them to be an important component related with proteasome activity. And having that known, we investigated all the 1433 molecules and identified that 1433 epsilon was one of the only gene that correlated, its expression correlated with sensitivity to both the proteasome inhibitor bortezomib and carfilzomib in a large panel of myeloma cells. And that was our starting interest, that looks like 1433 epsilon has some connection with sensitivity to proteasome. And we also observed that this proteasome response quite positively correlates with expression of this protein. So keeping that in mind, then we investigated how does this protein work and what does it do? So we found that this molecule, 1433 epsilon, promotes translation, initiation, and protein synthesis. And it does it through a very clear pathway, which is that we call TSC1, TSC2 complex, but better known is that it involves the mTORC1 or mTOR pathway. This is a pathway that has been described in myeloma previously, but not in regards to 1433 and not in regards as clearly about the protein synthesis, etc. And so what we found is that when we deplete this molecule, it causes 14% reduction in protein synthesis, and it decreases the intracellular abundance of the secretion of the light chain in myeloma. And so the whole picture comes together is the myeloma is a disease that produces a lot of protein, which is secreted, the immunoglobulin, and that's its function. So when we inhibit the proteasome, the protein burden changes, stresses the myeloma cell, leading to its death. That's how proteasome inhibitor work. Now, because this protein is involved in chaperoning of the protein, it is a clear connection with proteasome inhibitor. So when we overexpress this protein in myeloma cells, there is an upregulation of protein synthesis, and it increases protein load. And thereby, it can affect the sensitivity with the proteasome. That's what our findings were. The correlation was also observed in primary myeloma cells from two independent multiple data sets. And so when we came to finally clinical application, that when this protein expression is lower, the outcome in myeloma patient is lower. And this is the correlation that in patients who have 17p deletion, which is where this gene is located, this is also where p53 is located, that 17p deletion leads to lower expression of this protein, leading to having greater sensitivity, to having resistance, and it works through protein load. And we had some clinical data that confirm exactly this, that this relationship between the protein load is very applicable in regards to how we may utilize this eventually for clinical purpose.